Welcome back to Chapter 15. In the previous video, we were talking about the 4 stages of pathogenesis. Now we're going to look at the types of infection. As we saw in the previous video, the stages included exposure, also known as contact. Then the pathogen had to stick to the tissues or cells. Once adhesion was completed, invasion allowed the pathogen to spread into the tissues and evade the immune system. And then infection involves multiplication and tissue damage in the host. So after invasion, we have infection when pathogens are growing and multiplying and causing tissue damage. Infections can be local, that means they are confined near the portal of entry, such as Staphylococcus causing a boil on the skin, a urinary tract infection, or pneumonia that's localized to the lungs. A focal infection is spread from a local site to a secondary location. One example could be streptococcus from the gums, and you get an opening or a wound in the gums, allowing it to enter the blood and leading to a secondary infection. Systemic infections show widespread dissemination. So one example is varicella-zoster virus. So varicella-zoster virus can gain entry into the body through a mucous membrane, usually through the upper respiratory system. But then it can spread throughout the body, and this can eventually show as skin lesions in chickenpox. We also have primary infections. These can weaken the immune system, weaken your defenses, and lead to secondary infections. And examples include HIV that might compromise the immune system, allowing yeast to overgrow, which can lead to oral thrush. Or let's say you have the flu, right? So this is viral influenza that may weaken the respiratory system and lead to bacterial pneumonia. This checkpoint asks us to list 3 conditions that could lead to a secondary infection. So one could be immune weakening after a primary infection, such as getting bacterial pneumonia after flu. The second could be loss of normal microbiota balance due to antibiotic therapy. Let's say that someone is taking antibiotics for urinary tract infection that can kill off the bacteria and lead to overgrowth of yeast, and that allows perhaps vaginal yeast infections or thrush to develop. A third condition could be tissue damage or inflammation, and that could provide new entry points for pathogens to gain entry more easily into the body. For a pathogen to persist, though, it must also be able to exit the host and find a new one. This is known as transmission of disease from one host to the next, and our book shows a nice figure showing the various portals of exit as well. Common portals of exit, like we saw in that figure, include respiratory droplets, feces, urine, semen, vaginal secretions, and blood. Coughing and sneezing can release thousands of microbes into the air. Blood exposure can occur through insect bites or contaminated needles, and exit routes usually correspond to the pathogen's portal of entry. All right, let's recap 15.2. Our first learning objective asks us to summarize Koch's postulates and molecular Koch's postulates. So the classic set of Koch's postulates told us that the pathogen had to be present in every case of the disease and absent in healthy individuals. The pathogen, we had to be able to isolate it and grow it in pure culture, and then the cultured pathogen must cause the same disease when we inoculate a healthy host. Then we have to re-isolate that pathogen from the new host and make sure it's the same pathogen. In molecular Koch's postulates, we were looking at genes. So specific genes should be only associated with pathogenic strains. Inactivating that gene should reduce or eliminate pathogenicity, and restoring the gene would restore that disease-causing ability. So going back to regular or classic Koch's postulates, the significance was that these established a systematic way to link a microbe to a specific disease, but there were limitations, like some pathogens are found in healthy people, or some can't be cultured, or others don't cause disease in animals, such as HIV. In the significance of molecular Koch's postulates, these allowed us to identify genes that contribute to virulence, but genetic manipulation is not always possible for all pathogens. So not all diseases have suitable experimental models. For the second learning objective, explain the concept of pathogenicity or virulence in terms of infectious and lethal dose. We saw that pathogenicity is the ability of a microbe to cause disease, and virulence is the degree or severity of pathogenicity. And we measure virulence through the infectious dose, or ID50. This is the number of organisms or toxins, etc., needed to infect 50% of hosts. Whereas the lethal dose is the LD50. That was the number of organisms or toxins or viral particles needed to kill 50% of hosts. And a lower LD50 or a lower ID50 indicates a more virulent organism. For the next one, primary versus opportunistic pathogens. Primary pathogens cause disease in healthy individuals regardless of their immune status. And one example was Bacillus anthracis, which causes anthrax. Whereas opportunistic pathogens cause disease only when normal defenses are compromised or when they enter an unusual body site. For example, Staphylococcus epidermidis is normal on the skin, but it can cause infections if they grow as biofilms and enter through a catheter. Looking at the stages of pathogenesis, we saw exposure or contact, which is when the pathogen encounters the host through a portal of entry. Adhesion was also known as colonization, which is when pathogens attach to host cells using adhesins or biofilms. Invasion is when the pathogen spreads into tissues, hiding or evading our immune defenses. And infection is when the pathogen multiplies, producing toxins or tissue damage that cause the signs and symptoms. And then we have not quite a stage here, but remember the pathogen has to be able to exit and leave the host through a portal of exit to infect new hosts. Finally, looking at the roles of portals of entry and exit in the transmission of disease. So portals of entry are where pathogens enter the body. These are things like the respiratory tract, GI tract, the skin, or the parenteral route, or the placenta. Whereas the portal of exit is the route pathogens use to leave the host and transmit or spread to others, like through respiratory droplets, feces, urine, blood, saliva. So together, these portals determine how disease spread and persist in populations. Now let's transition to 15.3. Here we're going to focus on how microbes actually cause tissue damage through their virulence factors. So by the end of this section, we'll be able to explain how virulence factors cause disease symptoms, differentiate between endo and exotoxins, describe major exotoxin types, and explain viral adhesion and antigenic variation. Virulence factors are the molecules that give pathogens their disease-causing power. They help with attachment to host cells, invasion of tissues, evasion or hiding from the immune system, and damage to host cells. These genes are often grouped together on pathogenicity islands, which are clusters of virulence genes on a plasmid or on the main chromosome. If a key virulence gene is knocked out or silenced, the microbe becomes less virulent. Remember that after exposure, the next step was adhesion, and most microbes use structures called adhesins that are located on microbial structures such as fimbriae, pili, capsules, or viral spikes, or others, and they determine which tissues a pathogen can bind to. So some examples are shown in the table below from our book. Streptococcus pyogenes, that causes strep throat, for instance, uses protein F to attach to our respiratory epithelial cells. Neisseria gonorrhoeae, that causes gonorrhea, uses their type 4 pili to attach to our urethral cells. Vibrio cholerae has pili, so special structures on their pili that attaches to our intestines. And each of these adhesins targets specific receptors in the host, and that really explains host and tissue specificity. After adhesion, pathogens move deeper through tissues, and this is known as invasion. Many microbes spread by entering our blood, allowing them easy access to our cells and tissues. But the blood also contains the immune defenses. And there are a variety of terms describing pathogens in blood, depending on the type of pathogen. Bacteremia is when we have bacteria in the blood. Pyemia is pus-forming bacteria in the blood. Viremia, we're looking at viruses in there. Toxemia is toxins in the blood. And septicemia is when bacteria are present and multiplying in the blood. And this is dangerous, and it can cause a condition known as sepsis. So septicemia can lead to sepsis and then septic shock. In shock, blood pressure drops below 90 millimeters of mercury, which can deprive organs of oxygen. This is especially dangerous in gram-negative infections because these bacteria, remember they have that outer membrane, and they can release LPS or endotoxin. And endotoxin triggers immune cells to release tumor necrosis factor. Tumor necrosis factor increases the permeability of our capillaries, which leads to fluid loss, right? Fluid is going to leave the blood vessels to enter the tissues. That can cause edema or swelling, very low blood pressure, and eventually multi-organ failure if blood pressure is way too low and oxygen is not being delivered to tissues. And this is why sepsis is so dangerous. So it's really our immune system, our phagocytes, which release tumor necrosis factor when triggered by something such as endotoxin, and that increases capillary permeability. So here's normal, the normal state of the capillary, and you can see tumor necrosis factor can increase the opening between those endothelial cells, allowing fluid leakage and causing edema and all of those other signs and symptoms as well. Pathogens also secrete exoenzymes. These are enzymes that work outside of cells to help them invade, and they can facilitate invasion through our connective tissues, support the growth of the pathogen, and hide from or evade our immune system. One example is hyaluronidase S, which is produced by S. aureus, Streptococcus pyogenes, as well as Clostridium perfringens. This enzyme, hyaluronidase S, breaks down hyaluronic acid. This is kind of like a sticky cement type substance that typically holds our cells together in our connective tissues. And by digesting this material through the enzyme hyaluronidase S, the bacterial will loosen these cell junctions, allowing them to spread through the tissue layers and move beyond that initial infection site. This is why these infections can start small, but then expand rapidly if they're left untreated. Here's a look at that normal tissue here. We have hyaluronic acid, also known as hyaluronin. This is the same thing, that holds the cells together with the enzyme hyaluronidase, that is an exoenzyme that can break down those layers between the cells, allowing the bacteria to invade. Another type of exoenzyme is DNase, which is a type of nuclease, and DNase breaks down extracellular DNA around the infection site. It turns out that when host or bacterial cells die, they release DNA, and that forms a sticky net trapping microbes. Staphylococcus aureus, however, can produce DNase, and that will degrade or break down that mesh or net, helping these bacteria escape and spread. DNase can also break down another structure that's usually built by our immune system called neutrophil extracellular traps or nets. These are webs of DNA released by immune cells in order to trap bacteria. So DNase acts like scissors that cut through these immune barriers. Our next exoenzyme are phospholipases, and phospholipases are enzymes that attack the phospholipid membrane of host cells. Bacillus anthracis, for example, the cause of anthrax, produces phospholipase C, which destroys the phagosome membrane inside immune cells called phagocytes. This prevents the phagosome from fusing with a lysosome, and that lets the bacterium escape into the cytoplasm and multiply there. Other pathogens like Listeria monocytogenes or Rickettsia bacteria can use similar mechanisms as well. Phospholipases can also break down or lyse red and white blood cells, and that can contribute to further tissue destruction and inflammation. Pathogens can also produce proteases, And these break down host proteins for nutrition and invasion. They're usually classified, these proteases are classified by their target. For example, serine proteases target proteins that have the amino acid serine. Or sometimes we classify these proteases by whether or not they contain metals in their active site of the protein structure. For example, zinc metalloproteases, they contain a zinc ion, and that's necessary for the enzyme to work. A special group of proteases are the collagenases, and these digest collagen, which is a major protein in our connective tissues. Clostridium perfringens, for example, uses collagenase along with phospholipase and toxins to cause a condition known as gas gangrene. Here we have tissue necrosis and gas production, and this makes infections particularly dangerous because they can progress to septicemia if the bacteria enters the bloodstream. Our book provides a picture of a blood vessel with endothelial cells and a dense connective tissue shown in red, and we can see bacteria releasing collagenase, which allows them to invade and enter the blood. Here's a summary of the different types of exoenzymes and a few more pictures. Here, normally between cells, we have hyaluronic acid between cells, and we have collagen to protect from invasion. But if microbes release hyaluronidase, they can break through that hyaluronic acid layer and enter between cells. And then if they also release collagenase, they can break through that collagen layer as well to invade tissues and perhaps even enter the blood. Phospholipase C can break down our membranes. It can also allow microbes to break down the phagosome. So remember, the phagosome is the structure that is formed within a phagocyte, and usually it's going to fuse with a lysosome to digest the microbe. But if they can break down that membrane of the phagosome, then they can escape and replicate in the cytoplasm. So far, we've talked about enzymes that help bacteria invade. Now we're going to move on to the next category of virulence factors, and these are toxins. Toxins are biological poisons that directly damage host cells and interfere with cell function. The ability to produce toxins is known as pathogenicity, and that's what separates many harmless microbes from dangerous pathogens. There are two main types: endotoxins and exotoxins. So let's start with endotoxin. Endotoxin refers to that lipopolysaccharide layer, that LPS layer found on the outer membrane of our gram-negative bacteria. When these bacteria, these gram-negative bacteria, die or when they divide through binary fission, they release LPS into the bloodstream. The lipid A portion of that lipopolysaccharide is toxic, and it triggers inflammation by activating our immune system. At low levels, this can help fight infection by triggering things like fever. But at high levels, it leads to extreme fever, low blood pressure, and potentially multi-organ failure and shock. Endotoxin, unfortunately, is also heat stable, so it can survive most heating processes. Here we have the picture of endotoxin. This is that lipopolysaccharide layer with that lipid A portion being the toxic component that triggers inflammation and fever. The standard test to detect endotoxin is the Limulus amoebocyte lysate test, or LAL test, and it uses horseshoe crab blood. Horseshoe crab, this is the genus and species name. They have blood cells called amoebocytes, and when their blood cells, these amoebocytes, are exposed to endotoxin, it results in clotting formation. That indicates the presence of lipopolysaccharide, which therefore indicates the presence of endotoxin. And it's widely used in pharmaceuticals, medical device, and vaccine testing to make sure those products are free of endotoxins. So horseshoe crab blood is very interesting because you can kind of see it here. It has this natural bright blue color because this blood contains hemocyanin. This is a copper-based molecule. These days, there is another alternative that's not mentioned in the book called recombinant factor C, or RFC assay. This uses a synthetic version of the horseshoe crab clotting enzyme. It's called factor C, and it's able to detect endotoxin without using this animal source. So it's being used more and more for ethical and sustainability reasons, but unfortunately it's still less common than the LAL test. We also have exotoxins. Unlike endotoxin, exotoxins are proteins that are secreted by living bacteria, mainly gram-positive, but some gram-negative as well. They act in very specific ways, targeting certain cells or receptors rather than causing general inflammation. But because they are proteins, they are heat sensitive. They can be destroyed by heat, and they can also be converted to toxoids. These are inactivated toxins that can be used as vaccines. Exotoxins are also extremely potent. For instance, botulism toxin is hundreds of thousands of times more lethal than endotoxin by weight. All right, let's compare these side by side, endo versus exotoxins. Endotoxin comes from that outer membrane of gram-negative bacteria and can cause systemic inflammation. It's also heat stable. Exotoxins, in contrast, they're secreted proteins, mainly from gram-positive bacteria, that act on specific target cells, and they are heat sensitive. Endotoxins trigger things like sepsis and shock, whereas exotoxins produce precise effects like paralysis, diarrhea, or tissue necrosis, and this really depends on their mechanism. We also just saw that exotoxin has a low LD50, which means they are very potent compared to endotoxins. The first major class of exotoxins we have are called AB exotoxins, and these have two parts. The B is the binding subunit, which attaches to the host cell receptor. The A is the active subunit, which enters the cell and causes damage. So here we can see an image of that, the B binding to that host cell, and then they're taken in through endocytosis. Once they're inside that vacuole, A separates from B, and it gains access to the cytoplasm and can cause damage. Diphtheria toxin is a type of AB exotoxin. We can see the B component here, and the A component is going to inhibit the process of translation, and this is going to inhibit protein synthesis and thus kill the cell. Cholera toxin is another type of AB exotoxin, also known as an enterotoxin, as it results in damage to the GI tract. So what happens here is this causes increased secretion of cyclic AMP. That's going to disrupt the normal physiology of our intestinal epithelial cells, causing them to secrete excessive amounts of fluid and electrolytes into the intestines, resulting in severe diarrhea and water loss. The botulinum toxin that we see in botulism, actually before we do that, let's look at the normal physiology of muscle contraction, because that's going to be impacted when we have botulism. So normally the neuron, there is more detail to this, but we're going to really simplify it. To cause muscle contraction, the neuron is going to release neurotransmitters called acetylcholine that will bind to the muscle cell and induce contraction. So we're super simplifying this. In botulism, what's going to happen is what we see over here on that right side, that right panel. We are going to have the B subunit of the exotoxin bind to neurons and allow botulism toxin to enter the neurons at this neuromuscular junction. The A subunit is going to act as a protease, and that's going to break down proteins that are normally involved in the neuron's release of acetylcholine. So the result is that acetylcholine is not released and there is no muscle contraction. So botulism toxin or botulinum toxin results in flaccid paralysis, where you cannot contract your muscles. In tetanus toxin, this is also a group or inside the group AB exotoxins, these toxins are going to block inhibitory neurons. So usually when muscles are contracting, the agonist contracts and the antagonist muscle has to relax. And they relax because inhibitory neurons will release glycine and GABA to prevent the contraction of the antagonist muscle. But tetanus toxin will block this inhibition, allowing both muscles to contract at the same time, and that leads to spastic paralysis. So down here is what we see with tetanus toxin. Normally, if this is the antagonist muscle, let's say we're flexing our biceps, the triceps should be relaxed. And usually when you're flexing your biceps, or contracting, I should say, your biceps, the triceps will relax because GABA and glycine, these inhibitory neurons, will prevent the release of acetylcholine, so the triceps is relaxed in this state. But if tetanus toxin comes in, it's going to block that inhibitory neuron. There's no GABA or glycine released. Therefore, acetylcholine is being released onto that triceps muscle. At the same time, the biceps is contracting. The triceps is also going to contract, and that results in spastic paralysis. Up here we see that previous example of botulism toxin. This is more accurately known as botulinum toxin. So botulinum toxin is the more accurate way to note or name this toxin. It prevents the release of acetylcholine, so we cannot contract, and that was called flaccid paralysis. All right, we'll stop here for our third video and continue with our major exotoxin types in our next video.